Hello students, my name is Ho Jong Nam, and from now I'm going to have a very short time for introducing my lecture that is titled as Data Science in Healthcare. So this lecture is having code as EC6212, and this code has been code shared as AI6212. So first I prepared two cover from the Time magazine. In the left part, it shows that with the development of genetic engineering technology, we now can predict the susceptibility of certain diseases. So for example, after we are doing the genetic test, we can predict or know the high risk of cancer of ourselves or the other types of disease such as asthma and diabetes. And the right part of this slide shows the very famous story that the actress Angelina Jolie had a prevention surgery once because she confirmed that she is having very high risk of the breast cancers in her BRCA1 gene. So this kind of story tells that we are now facing the era of personalized medicine by analyzing our own genetic information. In this lecture, we are going to learn how we can analyze the large scale of genetic information in the viewpoint of data science. This lecture focuses on to understand the characteristic of molecular data, including genome and transcriptome and metabolome. And we are going to learn how we can analyze the data by learning the basic uses of the Linux system and the program languages and the basic statistics. And also we are going to learn the basic skills for discovering biomarkers for early diagnosis. And also we are going to learn the skills for the drug discovery for the treatment. This lecture is designed for students who are very interested in bioinformatics or computational biology or variety fields of the biology or medicine. And also the students who are interested in handling high throughput molecular data and analyzing large scale of the data. And students who wants to learn the use of the computation server and the data statistic and the concept of molecular high throughput data is very welcome. So let's start to preview the schedule, okay? The first part of the lecture is about the basics, okay? So we are going to learn the basic first. First, we are going to learn the Linux systems and the basic programming languages, including Python and R. And also we are going to learn the basics of the molecular biology information because the main data sources that we are going to handle in this lecture is about the genetic information that is starting from the DNA and transcribed into the RNA and translated into the protein. So this basic information flow and each characteristic of the molecular information is going to be detailedly explained. And also a bunch of the data sources that is having the genetic information is going to be covered. And the next part is about learning, which means understanding the data itself. So first, we are going to learn the technology, how we can actually read and understand our genetic information. And then we are going to learn how we can analyze or understanding the functional status of the cells using the transcriptomic data type, including RNA sequencing and the microarray. So after having the skills of the basics, then we are going to now jump into the analysis of the data. So we are going to learn the basics of the statistic to do the some types of the statistical test and having concluding some difference or this or the non-changes of the samples of the information. And then we're going to also learn the basics of the regressions and classification and the clustering to extract some important characteristic that the data itself is having. And because the cellular information is having very much interconnected format of functional aspect, so we are going to learn the graph-based of the interactive information handling part as well. So if you are having further questions, you can email me, and I hope to see you in this fall lecture. Bye-bye.